to British Biomedicine Institute. Today I, Dr. Pramod Khatri, is going to represent a simple topic named as clinical trial basic definitions. So what are clinical trials? Basically, these are the research studies on the subjects done at the hospital, and we try to answer the scientific questions and find a better way to treat the disease. So why are clinical trials important? So basically, the clinical trial, they translate result of the basic scientific research into the better way to prevent, diagnose or treat a disease. The most people take part, the faster we can get the answer of the critical research question, and we can also find the better treatment, and we can find the way to prevent a disease. Do many people take part in clinical trials? No, only a few people they participate, because clinical trials, they have tight inclusion and exclusion criteria. What are the different types of clinical trials? So basically, they are divided into 5 types, like treatment, prevention, early detection or screening, diagnostic trials, or quality of life or supportive care. So now comes treatment trials. So basically, what new treatment can help people with a particular disease? What is the most effective treatment for people with that disease, like COVID? So clinical trial phases, they are basically phase 1 to phase 4, and phase 4 is known as PMS, post-marketing surveillance. So here, phase 1 trials are basically done on the smaller amount of populations or subjects, and we want to know the agent effect on the human body, and we also want to know whether the dosage which has been given to the patient is safe or not. Phase 2 trials are basically done in 100 to 500 subjects, and here we want to know whether our new agent or intervention have an effect on the disease or not. Phase 3 trials, they are basically carried out in 800 to 1000 subjects, and here we want to know whether the new agent or intervention or new use of the treatment is better than the previously available medicine over the counter, and we also want to know whether the participants or subjects who they have an equal chance to be assigned either one or two group of medicines, like one group can get main medicine or active medicine, another group can get a placebo or a best available therapy. So here the main objective of phase 3 is to know that the medicine which has been prepared by the sponsor right now is superior to the medicine which is available over the counter. So randomized trials are basically those trials in which the participants, they are not aware about the assigned group, whether they will be receiving the main medicine or active medicine, or they will be receiving the placebo. So basically, this is the most widely used technique or design in the clinical trials, particularly in phase 3. So here the participants, they have equal chance to be assigned to one of the 2 or more groups, and one get most widely accepted treatment, or we can say standard treatment, and the host get the new treatment being tested, which researcher hopes and have the reasons to believe will be better than the standard treatment already available in the market. So randomization is basically a step, as it is visible in this photo. So here we are randomizing a group of people. If suppose there are 10 subjects, then here 5 patients, they will get main medicine and 5 will get placebo medicine. So why is randomization important? Basically, so all groups are alike as possible, because here every patient, they have an equal chance of getting the active medicines, and with the help of various techniques which are available, we can randomize a patients, and it also provides the better way to prove the effectiveness of medicine in a clinical trial. So most of the trials are now randomized. Now comes the treatment trials. Treatment trials are basically what new treatment can help people with particular disease. What is the most effective treatment for people with that disease? Like now you are aware of COVID vaccine. They are now effective treatment for coronavirus outbreak. In the treatment trials, placebo are almost never used. Placebo is used only when no standard treatment exists. Patients are told of this possibility before deciding to take part. Prevention trials are basically used to evaluate the effectiveness of way to reduce the risk of a particular disease. And we only enroll healthy subjects who are at higher risk of developing that particular disease. So prevention trials are basically divided into two types mostly: action studies and agent studies. Action studies are like doing something. Agent studies are like taking something, or also called chemoprevention studies. So basically chemoprevention trials are those trials which compare the promising new agent with either your placebo or with your standard agent. So clinical trial protocol is basically a blueprint of, or we can say a recipe, and basically it consists of strict scientific guidelines like purpose of the study, how many patients we will recruit, who are eligible to participate like inclusion exclusion criteria, how the study will be carried out, what information will be gathered about the participants, on whether the participants they will be getting their diagnostic test or hematological test, and the endpoints. They are clearly visible in the protocol. So now comes the benefit of participations. The possible benefits for the patients are: patient will receive at a minimum the best standard treatment. If the new treatment or intervention is proven to work, patient may be the among to benefit from that first. Patient also have a chance to help other and improve their patient care. Risk of participations are basically new treatment or interventions under study are not always better than the standard care. Even if new treatment has benefit, it may not work in every patient. And health insurance and managed care providers, they do not always cover a clinical trial. So now comes patient protection. They have unfortunately been past abuses in the patient protection. I hope you have seen the history of clinical research in our previous YouTube video. And federal regulations ensure that people are told about the benefit, the risk, and the purpose of resource before they agree to participate in any of the clinical study. How are patient rights protected? So basically here, one of the most important right is being protected by the Institutional Ethics Committee or Institutional Review Board of a hospital, and informed consent is given to the patients. They can decide, once they have received all the information pertaining to the protocol, they can decide whether to participate or not to participate. Scientific reviews, scientific committees are available, and even DSMB is also available, a Data Safety Monitoring Board. They all review the documents from time to time. So how are patient rights protected? So basically, patient rights are generally protected with the help of informed consent. So first, the PI will be giving the information to the patient who is eligible for the study, and then patient will decide whether or not to participate. And if he has signed the informed consent, patient can skip, patient can get out of the study at any time. There is no burden. Okay, once you have participated, once you have signed the informed consent, you will participate till end. No, patient can skip or patient can get out whenever he want from the study. So how are patient right protected? Like there, I told you, there are scientific reviews, institutional ethics committee or institutional review board are there in the hospitals, and basically they helps in protection of human subjects from any type of injury. So how are patient right protected? We can use DSMB, Data Safety Monitoring Board. They ensure that the risk is minimized, advantage is high, medicine is safe, ensure data integrity, and stop a trial if a safety concern arises or objective is not met in between the trial. How do Why do so few people participate in clinical trials? So basically, here first point is that they are not aware. They do not have access to clinical trials and they may be afraid or suspicious of research. They cannot afford to participate and they may not want to go against healthcare provider wishes like your insurance. Healthcare providers might provide lack of awareness of appropriate clinical trials, be unwilling to lose control of a person's care, believe that standard treatment is best, be concerned that clinical trials add administrative burden. So, where to find clinical trial information? Basically, we can get information from the websites, government websites, or NLM, National Library of Medicine, or through your healthcare provider. So now comes the common definitions in the clinical trials. Adverse reaction. Basically, it is an unwanted effect which has been caused by the administration of a drug, and onset may be sudden or it may develop over a period of time. Approved drugs are those which has been approved by the US FDA and sponsor have carried out various phases like phase 1, phase 2 and phase 3, and they have received the approval from FDA and now they can market that medicine. And basically the approval process involves several steps including your preclinical laboratory or animal studies, clinical trials on healthy subjects for their safety and efficacy, filing a form of like your NDA or the new drug application or your IND, investigational new drug application to the FDA, which will be reviewed by the FDA, and once FDA can either issue acceptance letter or rejection letter based on the application which are being submitted by the sponsor. Basically, any of the treatment group in this randomized trial, most randomized trials have 2 arms, but some have 3 arms or even more. Baseline. The information which has been gathered from the patients at the beginning of the study, and basically we want to know values or quantity with which an unknown is compared with when measured or assessed. Bias. So in the clinical trials, it is important to remove the bias of any kind. So when a point of view prevent impartial judgment on issue related to the subjects of the point of view. In the clinical trials, bias is controlled by blinding and randomization. Blind. Here means key, if suppose it is a double blind, it means that the patient and the doctor, they are not aware. If it is single blind, it means patient is not aware whether he is getting main medicine or placebo. So basically in randomized trial, a blind happens if a patient is not told of which arm he is participating. A clinical trial is blind if participants, they are not aware on whether they are on the experimental drug or on the control arm of the study, and hence they are masked. Clinical investigator, or we can say principal investigator or PI, he is the main person who is controlling the main clinical trial and he mostly moves with the protocol rules and regulations. Clinical trials. Basically, clinical trials is a research study to answer specific questions about vaccines or new therapies or a new way of using treatments. Clinical trials, also known as medical research or research studies, are used to determine whether a new drug, treatment are both safe and effective, and we carefully conduct the clinical trials based on the protocol requirements. Cohort. Basically, in the epidemiology, a group of individuals with some characteristics in common are known as cohort. CBCT. Community-based clinical trials are basically conducted primarily through a primary care physician rather than academic research facilities. Compensate use is basically a method of providing experimental therapeutic prior to final FDA approval for use in humans. The procedure is used with a very sick individual who have no other treatment option. Often, case-by-case approval must be obtained from the FDA for compensate use of a drug or a therapy. Confidentiality regarding trial participants. So basically, all the information which will be gathered in accordance with the protocol, which will be available in the source note of that patient, must be confidentially hidden. So basically, it refers to maintaining the confidentiality of trial participants, including their personal identity or all personal medical informations. The trial participants consent to use the record for data verifications. Purpose should be obtained prior to trial, and assurance must be given that confidentiality will be maintained for the patient's document. Contraindication, basically a specific circumstances when the use of certain medicines can be harmful. Control group is basically the standard by which the experimental observations are evaluated. In many clinical trials, one group of patients will be given experimental drug or treatment, while the control group is given either a standard treatment or for the illness or a placebo. So now comes DSMB, Data Safety Monitoring Board. Basically, it is an independent committee which composed of community representative and clinical research experts, which will review the clinical data which has been generated from the patients in timely manner. And DSMB may recommend that a trial may be stopped if there are safety concerns or if the trial objective is not achieved. Diagnostic trials are basically those trials which are conducted to find the better test or procedure for diagnosing a particular disease or a condition. Diagnostic trials usually include people who have signs and symptoms of a disease or condition being studied. Dose ranging studies are those basically in which two or more doses of a drug are being tested in order to determine which dose will work best and which one is harmful. So here basically we will be increasing the dose range in the population. Suppose a patient will receive first 0.1 mg, after 1 week 0.2 mg, after next week 0.3 mg. So here we want to know the maximum tolerable dose or minimum tolerable dose. Double blinding studies, basically doctor and patient, they are not aware of which group they are assigned. So a clinical trial design is whether neither the participating individual nor the study staff know which participant are receiving the experimental drug or which one is receiving the placebo. Double-blind trials are thought to produce objective results since the expectations of doctor and the participants about the experiment drug do not affect the outcome. Also, they are also known as double mask study. So mostly they are used in the randomization trials. Drug-drug interaction. Basically a modification of effect of a drug when it is administered with another drug. The effect may increase or decrease. or it may also produce adverse effect, signs and symptoms which are not normally associated with either drug. Efficacy. Maximum ability of a drug or treatment to produce the result regardless of a dose. The dose pass efficacy trial if it is effective at the dose tested and against the illness for which it has been prescribed. In the procedure mandated by the FDA, the phase 2 clinical trials goes efficacy and phase 3 trial confirm it. Eligibility criteria is basically found in the protocol which specify which patients will be included and which patient will be excluded from the study. So it is basically a summary criteria for participant selections like your inclusion and exclusion criteria. Endpoint. Overall outcome that protocol is designed to evaluate. Epidemiology. Basically, it is a branch of medical science which deal with study of incidence and distribution and control of a disease in a given population. Experimental drug. A drug that is not FDA licensed for use in humans or as a treatment for a particular condition. FDA. So FDA is basically a US Department of Health and Human Service agency which is responsible for ensuring that the safety and effectiveness of drug, biologics, vaccines, medical devices, including those used for diagnosis, treatment, prevention of the disease like HIV, AIDS. The FDA also works with blood bank industry to safeguard the nation's blood supply. And we can get more information on their websites named as www.fda.gov. Hypothesis. Basically, it is the supposition or assumption advanced as a basis for reasoning or argument or as a guide to experimental investigations. Now comes inclusion and exclusion criteria. So basically, the medical or social standard determining whether a person may or may not be allowed to enter a clinical trials. These criteria are based on the factors like age, gender, type and the stage of a disease, previous treatment history and other medical conditions. It is important to note that the inclusion exclusion criteria are not used to reject people personally, but rather to identify appropriate participants and keep them safe. Informed consent. Basically, it is a process of learning the key fact about a clinical trial before deciding whether or not to participate in a clinical study. It is also a continuing process throughout the study to provide information for participants to help someone decide whether or not to participate. The doctor and the nurse involved in the trial explain the detail of the study and then patient can decide whether he is interested or not. Informed consent document. A document which describes the right of a study participants and it includes details of the study protocol like a purpose, duration, required procedure and the key benefit. Risk and potential benefits are also explained in the informed consent. The participants then decide whether or not to sign the informed consent, and if informed consent is not a contract, and the person may withdraw from the trial at any time whenever he wants. Institutional Review Board IRB or Institutional Ethics Committee IEC. So basically it is a committee of physicians, statisticians, researchers, community advocates and others that ensure that a clinical trial is ethical, that the right of the study participants will be protected. All the clinical trials in the USA must be approved by the ethics committee before they are started. Every institution that conduct or support biomedical or behavioral research which involve human participant must by federal regulations have the IRB that initially approves and periodically review the research in order to protect the rights of the human who are participating in a clinical study. NDA, new drug application. Basically, it is an application which is being submitted by the sponsor to the FDA after the clinical trials have finished, particularly your phase 3 trials, and for a license to market for a specific indications. So before carrying out the PMS or phase 4 trial, NDA application will be submitted by the sponsor to the FDA for marketing their medicine. Off-label use, basically a drug which describes the condition other than those approved by the FDA. Open label trials are basically everyone is aware, doctor, patients, they are aware which treatment patient is getting. A clinical trial in which the doctor and the participant know which drug or vaccine a patient is administered. Orphan drugs, basically FDA agency that refer the medications used to treat disease and conditions which occur rarely. There is a little finance incentive for the pharmaceutical industry to develop medications for these disease or conditions. Orphan drug status, however, give a manufacturer specific financial incentive to develop and provide such medicines. Pharmacokinetics is basically a process in which we study ADME, absorption, distribution, metabolism, and excretion of the drug or a vaccine. Phase 1 trial are the initial studies which are used to determine the metabolism and pharmacological action of a drug in the humans, the side effects associated with increasing the dose, and to gain early evidence of effectiveness generally include healthy participants, which can range from 50 to 100. Phase 2 trials are basically the controlled clinical trials which are conducted to evaluate the effectiveness of a drug for a particular indications in patient with a disease or condition under the study, or to determine the common short-term side effect and the risk which are involved. Phase 3 trials are basically the expanded control or uncontrolled trials after preliminary evidence which suggests the effectiveness of a drug has been obtained, and they are intended to gather additional information to evaluate the overall benefit risk relationship for the drug and also provide adequate basis for physician labeling. Phase 4 trials are also known as PMS, post-marketing surveillance or studies. Basically, they are used to get additional information of the labeling of a drug, like your risk, your benefit, and optimal use in a wider population. Placebo is basically a dummy medicine, or we can say it is an inactive pill, liquid or powder that has no treatment value. There are no active pharmaceutical ingredient, API. In clinical trials, experimental treatment are often compared with placebo to assess the treatment effectiveness. Placebo effect, because this is a nil medicine or dummy medicine, the physician or emotional change can happen after a substance is taken or administered that is not the result of any specific property of a substance. The change may be beneficial, reflecting the expectations of the participant, and are often the expectations of the person giving the substance. Preclinical, or we can say animal study. They refers to the testing of experimental drugs in a test tube or in animals. The testing that occur before trials in human may be carried out. Prevention trial basically refers to a trial to find a better way to prevent a disease in a people who have never had a disease, or to prevent a disease from returning. These approaches may include medicines, vitamins, vaccine, minerals, or lifestyle change. Protocol, basically it is a study plan on which all clinical trials are carried out. The plan is carefully designed to safeguard the health of a subject as well as answer scientific specific research questions. A protocol describe what type of patients will be included in a trial, schedule of the test, the procedures, medications and the dosage form, and the length of the trial. While in a clinical trial, participants follow a protocol or are regularly seen by the research staff like your staff nurse, your coordinator or sub-investigator in order to monitor their health and to determine the safety and effectiveness of the treatment. Quality of life trials, QOL. So basically they are the trials which explore the way to improve comfort and quality of a life for an individual with a chronic illness. Randomization is basically a method based on a chance by which the study participants are assigned to a treatment group. Randomization will minimize the difference among the group by equally distributing people with particular characteristics among all the trial arm. The researchers do not know which treatment is better from what is known at the time, and any of the treatment chosen could be the benefit of the participants or subjects. Randomized trials are basically a study in which the participants are randomly picked up or they are assigned one or two or more treatment arms. Or they are basically occasionally we use placebo. Screening trials basically refers to trials which test the best way to detect certain disease or health conditions. Single blind studies are basically those in which the patient is not aware, in which one party, either the subject, either the investigator or participant, is unaware of which medication the subject is taking, and they are known as single mask study. Standard treatments are basically the treatment which are currently available or which are widely used, and they are basically approved by the FDA, and they are considered to be effective in the treatment of specific disease and condition. Toxicity, the adverse effect which has been produced by a drug that is detrimental to participant health. The level of toxicity which has been associated with a drug will vary depending on the conditions with which the drug is used to treat. Treatment trials are basically refers to the trials which test new treatment, new combinations of drug or new approach to surgery or to radiation therapy. Persons which are involved in the clinical trials are basically sponsor, CRA, investigator or sub-investigator, monitor, CRC, auditor, clinical data manager, project manager, statistician, and ethics committee, local institutional review board or local FDA, a country specific FDA. These are the key persons which are helping in running a smoothly clinical trial at a hospital. So this is all about the presentation. I hope you have gained a lot of clinical information through this presentation. Please like, share, subscribe our YouTube channel named as British Biomedicine Institute. Good luck, goodbye.